Yeah. Yeah, perfect. So we'll wait another couple of minutes, and then I don't know whether everyone can hear us or not. This is, yeah, Jenny saying, please hit record. Okay, we can hear you. Okay, so you have to be careful when you start these because everyone can hear the preamble. Okay, give it one more minute and then we'll start. Yeah, Stephanie, if you want to share your screen. Okay, so welcome everyone to this month's seminar. The introduction to the seminar, I had asked Steve Reese to give it because I think he's much more appropriate on this subject because he's the expert here at Pitt and UPMC. Before starting, I would just say that there'll be notes going out. Next month will be Elaine Mardis speaking about the genome center she set up at Children's at Cincinnati. And then the month after that will be Heidi Rehm from Harvard and Mass General is going to talk about her population genomic studies. I would also say, some of you may have seen this, we have the Precision Medicine World Conference downtown in the Wyndham next week. We are pretty much sold out for attendees, but if you want to go, you could still register as a last minute, and it will have a session on, of course, population genomics, and then there'll be sessions on data science and a number of other areas, the neonatal sequencing programs going on from the 4 main centers in the US, and a bunch of other exciting precision medicine topics. There will also be a meeting on pharmacogenomics run by Phil Empey on Wednesday, which is also at the Wyndham downtown. If you search your emails, those messages have been going out routinely. So I'm happy to transfer this over to Steve Reese, who was the one that suggested to have Steph Devaney speak, and will introduce her. Thanks, Steve.
All right, thanks, Adrian. It's an honor for me to introduce Dr. Stephanie Devaney. Dr. Devaney is a molecular geneticist by training who spent much of her career focusing on research policy. She spent time in the office of the director at NIH, shaping research policies, but also involved in strategic planning for NIH's vision in several areas, including precision medicine. And from there, she moved on to the White House, which is, Steph, I think that's where we first met, when you were in the White House, where she was a key leader in President Obama's precision medicine initiative, which eventually evolved into the All of Us research program. Today, Steph is now the COO of the All of Us research program, and she's going to give us an overview on All of Us and precision medicine research. So, Steph, thank you so much for joining us today.
Great, thank you very much. I want to start by thanking you, Steve, and Dr. Lee for inviting me to join and talk about the All of Us research program and our mission, which I'm very excited about. I know you are, and hopefully by the end of this talk, everyone else will be as well. My only regret, of course, is that I can't be there with you all in person and see your faces and interact in the way that I think we all miss at conferences after the talk and during the talk and all of that. But we will take on the Zoom challenge. And I'll start by also mentioning that I have a cat who is delightful and quiet until I'm giving a talk or having a serious conversation, and then we'll be very disruptive. So this is my current setup right now, just in case something wild happens in the background, it's most likely my cat. So let me start with the mission of all of us, which is it really was at the core, and Steve mentioned that, you know, all of this work really started as a collective of government agencies really invested in the idea that we could ultimately end up with individualized prevention, treatment, and care for all of us, and that by putting real infrastructure in place to allow health research with all of the brightest minds across the country, that we could meet that mission. So the All of Us program, it has a mission that we share with a number of other organizations in the broader ecosystem of precision medicine, but we have some pretty unique attributes to the program, including the way that we think about our relationship with participants. And in the long history of research in which participants have often been the subject of research, and data has been taken from them and used for research, we really have thought about this in a much more dynamic way. Our emphasis here is on building partnership with our participants. And that goes for participants from all walks of life. And I'll talk a little bit more about our priority around diversity here a little bit later on. And then to take the data that our participants have very generously shared with us and deliver one of the largest, richest biomedical data sets that's broadly accessible to a diverse set of researchers to access and ask all sorts of questions across the health and disease spectrum. And ultimately, that data and the specimens and the recontactability of our participants, that then will create what we hope is a real robust ecosystem of researchers and funders and folks who can build on top of the platform and support the use of it, and ultimately we can expand the amount of information that we're gleaning from the program. I mentioned diversity briefly. I will say that from the very beginning of the program, the very, very beginning from day one, I mean, Steve and I were having conversations way back when I was downtown, and we were at that point before we had a single dollar appropriated to the program. We were talking about the real transformative aspect of this program is going to be diversity, and really reflecting the diversity of the United States and overemphasizing or over-enrolling participants who have been underrepresented in biomedical research for various reasons. And that was really going to take building partnership with participants and showing that we could be trustworthy and that we could understand what individuals from all communities across the country really would like to get out of a program like this. The program is longitudinal in design, so our mission is to capture all sorts of data from participants over the course of their life. And the idea is that as somebody goes through life and stays healthy or gets sick and then gets healthy again, and whatever happens to them over life, that really the true value and the richness of the data will come as the data gets more and more longitudinal, and we can sort of see how people experience life and the effect it has on health outcomes. We think about the data types also in terms of diversity. Our goal from the beginning was never to create a genomics program. It was always to create a program that could start to think about environment by lifestyle, by biology. And so we are really focused on capturing a whole bunch of different types of data, including genomics. That includes electronic health record data that's captured over the life course, surveys that people report themselves, so participant reported information through surveys, some physical measures that we capture at the very beginning of enrollment, and then biospecimens so that we can run genomic assays as well as others in the future. I mentioned the accessibility of the data on the research side, and I'm excited to spend some time today talking about this with you all. It was just noting that we have spent so much time in the early years of the program thinking about the infrastructure and the enrollment side of things. And we're really excited to have opened up the data, the first sets of data from our participants. So the idea here being that the data needs to be open to all. We are not a program where you have to be affiliated with our institution or collaborate with someone from our institution. That the idea here is that really this is a national resource, and that therefore it should be open to all researchers wherever they sit. And then doing all of this with security and privacy at the front of our minds, and making sure that the data that people provide is well cared for and protected. So we do all of this with just a huge number of partners from across the country. It truly is a national network of folks from hundreds of different institutions. And you can see here some of the different partners that we have, including the enrollment sites, and those are healthcare provider organizations. We've got federally qualified health centers involved. We've got the VA and different VA sites involved. And then we've got a number of partners who help us think about community engagement and communications, and how we really get to the interests of individual communities, both locally and nationally. And then all of the partners that help us build out the data resource, so where the data lives and who can access the data and how you access the data and running the analyses. We keep all of our data centrally located in the cloud, so it's not being dispersed to researchers. And so that requires a lot of organization on the side of our technology partners. And then of course, we've got genomic centers that are helping us run 1 million whole genome sequences, which is a heavy mandate. So we've got 3 genome centers. So this truly is a partnership across the country, unlike very few others. And we're really devoted and invested in building out this network of partners, because I just think we have the savviest, most sophisticated and caring and mission-driven set of partners that one could hope for. And included in that is our friends here at University of Pittsburgh under the leadership of Dr. Steve Reese and the team that he's been leading over the years, and helping to enroll participants from the Pittsburgh area and from many other areas now with different clinic sites or enrollment sites, and really getting into different communities in and around Pittsburgh and helping participants join the program. So University of Pittsburgh has been a partner from the very beginning, and it's been really nice to shape the program together. So it's especially an honor to speak to you all today. So where are we now? I mentioned that we have been enrolling participants and really have been focusing for the first few years on participant enrollment and building all of the necessary infrastructure to do that. We have about 408,000 participants that have consented to the program, and about 296,000 of those have completed the initial steps of the program. I'll talk a little bit more about that. We've got a little over 250,000 participants from whom we are collecting electronic health record data, and in our biobank at the Mayo Clinic, we have over 300,000 biosamples from participants. And you can see on the right, we started enrolling back in May of 2017 in a very sort of early alpha beta phase where we were testing enrollment, and the national launch happened in May of 2018. And so you can see a little uptick there. And then of course, for the rest of our lives, all of us as humans, and those of us working in this program and others, we will always have that dip when the COVID-19 pandemic took over our lives, and we stopped with the in-person enrollment for the sake of safety for everybody. And we're starting to do some of that again, the in-person enrollment, but it has also required us to be creative about how we do enrollment with participants. And I think there's a real future here in thinking about how participants join research studies and how we engage with them over time. So in some ways, it has sort of forced our hand in getting more flexible on how we do enrollment. And here's a little bit more on the breakdown, just to note that we are extremely proud of the fact that we have been able to overrepresent folks that have been underrepresented in biomedical research in a real way, with over 80% of our participants from groups that have not been represented well. And we are not currently enrolling children, although we're really excited to start pediatric enrollment over the coming years. You can see we've got folks from all age groups over the age of 18. And happily, we're at about 53% plus racial and ethnic minorities. And coming from the genomics discipline and knowing that most of the genomic data sets have been largely European for a very long time, this is something we're especially excited about when we can get really representative or reflective in an important way around diversity. So just quickly, I want to just share with you all how the different steps of enrollment for a participant joining the program. We do ask participants to consent electronically, and you can do that. They can do that from anywhere as long as they have internet access. It's an online consent. And we ask them at that time to share to authorize us to share their electronic health records with the All of Us research program. And that's in partnership, like, for example, with the University of Pittsburgh. We then capture the electronic health records on those participants at a regular cadence. And that is now shaping up to be a nice longitudinal data set. In addition, we ask people to share a bunch of information about themselves that really only they can report in certain ways. And that's through surveys. We have a number of surveys capturing demographics and lifestyle of our participants, personal and family medical history, healthcare access, and then we did launch a survey around COVID-19 that was really geared around participant experience and covered all sorts of things like economic stress and loneliness and some of the features of COVID-19, whether you became infected or not, that it really did drastically change people's lives. We're very excited to launch our next survey later this year on social determinants of health, where we can start to gather information about people's lives that certainly do have an effect on health outcomes. And part of the test is to figure out how. But as a program that is so invested in diversity and health equity and health disparities, it's really important that we gather information about people's social support, loneliness, food insecurity, perceived stress, daily spiritual experiences. You can see all the different types of information we're hoping that folks will share with us so we can start to look at the effects that those things might have on our overall health. And then we do capture physical measures as someone joins the program, as well as biosamples through blood, saliva, and urine. As I mentioned earlier on, with the start of the COVID-19 pandemic, we got even more creative around how we're doing sample collection. And so the saliva collection kit has now become a thing that we're using more and more in the program, so that folks who don't want to come in for a blood sample, can't come in to give a blood sample, can still participate in the program. We can collect their saliva through the mail and generate DNA or genomic sequence data from that. And then finally, we do have the ability for participants who want to bring their own device, so they can connect their Fitbit or their Apple Health Kit to their device, and that data then will come directly into our data set, and then it can be connected to the rest of their data. And so we're hoping to learn some interesting things there about that sort of passive data collection and what it can tell us about participants and their health. So as a corollary to this, I just talked about all of the things we're asking our participants to share. I just want to take a moment to talk about value, because we, as I mentioned before, we're really not interested in a model where we just take data from people. We really want this to be of value to participants in return. And I think this is an understudied topic, and we have a lot to learn about what real value might look like to participants, but how we can really change the paradigm of participating in research studies. One of the ways that we do that, and one of the ways we think about value, is in the return of information and making sure that participants have access to their information. This slide shows the core values that have been instituted in the program from day one, and we we do use these as we make decisions about the program because they are important to us, and participants having access to their information is not always the easiest thing. In fact, it's never the easiest thing to do. But by stating it as a priority, we have really held ourselves to that, and we're already returning genetic information to participants and other types of information, including comparative survey data. And there's a real opportunity here as we get more and more science being generated on the data to share with participants what's happening with their data in terms of aggregate results, scientific findings, other things that are coming out of the use of their data. So both individual level information about themselves, and then also how the data is being used. I think there's a ton of room here to get really creative and understand what might motivate participants to stay engaged in a study like this. So as I mentioned, we have been returning genetic information to participants. We've started with ancestry and traits. And so we have a few traits that we will return to people who want to get that information back, as well as their genetic ancestry. And that has been a good opportunity for us. We started with non-health related results, allowing us to both understand the value of this for participants, as well as helping us to get good at communicating genetic information to participants before we get into really the high risk stuff. And we're right now gearing up to return health related results to participants starting next year. So our goal around health related results is, and we have gone through a regulatory approval with the FDA, and we have approval to return the ACMG genes, and we're actually working off of ACMG 59 list still, and I know they've made some updates there. As well as pharmacogenomics. So once we are up and running with that early next year, participants will have the choice to say, yes, I do or do not want to learn about my any health-related genetic information. And if they choose to, then we will have that interpretation done at a CLIA lab so that we know that it's valid. And the results will be returned through a genetic counselor so that our participants, especially if learning about a hereditary disease risk, will have the support of a counselor and won't be left to, just read on their own. So we're really anxious to get that up and running and learn a lot about how that communication with our participant works. So now I just want to switch to researchers, because this is the area where we've just really started to turn our attention recently, as we launched the first data set in May of 2020. So I am looking at our core values again, and I mentioned this a couple of times. This resource is really meant to be broadly accessible for research purposes. And from our perspective, that means researchers from all sectors, from both within the United States as well as outside of the United States, from all different types of institutions, and including community researchers. And so we have had some limitations around who has access during the beta phase, but over time, we really want to expand to a world in which the data truly is broadly accessible to folks with a valid research question. So this is busy, but what I want to point out, and just want to walk you through the different types of data that are now available, especially for those of you who want to take a look at data immediately. We have a public tier, which we call our data browser, which is at databrowser.researchallofus.org. You can see the link there on the bottom. That is a public data set. There's no login required. It's all of aggregate data counts on our participants, and it looks something like this. So you can see summary statistics of our participants, including EHR data, the survey questions, physical measures. And when you go in there, you can just sort of play around with different queries. For example, if you were to run a search on dementia, and you get any EHR conditions that mention dementia, and you get any survey questions that specifically relate to that coming up there on the bottom, and you can then see things like the top 10 concepts that relate to dementia and participant counts. And you can also then pull out, if you wanted to pull out, say, concussion and dementia, you can look at those by age of diagnosis. We are careful on the public data browser to not allow for too many crosstabs because of privacy. And so if you really want to get into more specifics of the breakdowns, that's when you have to register for access to the row level data. But the public data browser is a great place to start if you want to look and see if the condition that you're specifically interested in is present and at what levels and what types of data we might have related to that. So I'm just going to switch now to the individual level data, because this is where the real analyses can happen. We have what we call a register tier, which is open now. We launched this in May of 2020, and it's cloud-based. As I mentioned, all of the data lives in the cloud, and researchers have to come to the data, and we've got tools and we've got everything there, tools and support there for a researcher. So we don't allow the data to leave our system or be downloaded. All of the personally identified information is removed from the data at that point. This is called our researcher workbench. The individual projects don't need IRB approval. I know that that's sort of dependent on the individual institution, but all of the data has been de-identified. And we use what we call a passport model, where an individual user who gets approved to access can go in, create as many workspaces as they want to, depending on how many questions they have. They have to write a description, and we make all of the data use descriptions public, because we are deeply invested in the idea of transparency, and we want our participants and everybody to be able to see what types of queries are going on in the workbench. But then you can start running your analyses. I mentioned that during the beta phase, we had some access limitations. We started with only folks who have an ERA Commons ID, which is a thing that not all researchers have, but it allowed us to do ID verification. We're working now on changing those requirements so that we can continually open and expand and make it more and more accessible, because that's ultimately our goal. You can find all of this at researchallofus.org. There's a ton of resources there to learn more. But essentially what happens is when you go in, I'll talk a little bit about the access process in a minute, but when you do get approved to create a workspace, you can build, you generate a workspace, and here's where you have to, say what you're going to do. You can build cohorts and data sets using tools that we've got, including Cohort Builder, Data Set Builder, and then run analyses using Jupyter Notebooks. So we have all of those tools available there, and really good support through the Data and Research Center at Vanderbilt, who works with Broad and Verily in building and maintaining this environment and supporting it. Currently in that data set, that data that's available to researchers on the researcher workbench, there's about 329,000 of our participants that are represented there. And some of the participants, we sort of froze the data at one point. So some of the more newly enrolled participants, their data is not yet in there, but will be added over time. We've got 214,000 plus electronic health records, 329,000 surveys, and then even over 11,000 Fitbit records, which we're excited about, because that really is bring your own device at this point. And I just want to show you, because we started enrolling people really at scale in 2018. It would be easy to think that our data set sort of starts in 2018, but in fact, because of the very rich electronic health records that we've been able to capture from participants who've agreed to share them in connection and collaboration with our partner healthcare provider organizations like the University of Pittsburgh, we're really able to look back in some cases on participants decades. So you can see some of our data even starts back in the 1980s with more and more dense data, of course, up closer to when we launched, and you can see the program launch date right here. The nice thing here as well is that while I mentioned we haven't started enrolling children yet, we do have some pediatric records here because of how far some of these records go back and how long some participants have been with their current healthcare provider. So just a quick snapshot of who's currently using the data ever since we launched it in beta. Since our launch in May, we have about 1,000 or a little over 1,000 registered researchers with 660 active projects. And we've already got some publications coming out of those uses. So that's been nice to see, because ultimately we're interested in the outcomes here. We have over 240 registered institutions, and I'll come back to that in a moment, but you can see most of the states here are represented in terms of having researchers registered with the data set. And over 24% of the institutions are historically Black colleges and universities or Hispanic serving institutions, which is great for us because we're excited to promote diversity on both the participant side as well as the researcher side, believing that that we really need all bright minds engaged in answering questions and asking questions of these data. Here's just a little bit more of a breakdown to show that most of the analysis that are going on right now are disease focused, but there are some other things, including methods development, public health research. So you can see the different types of research projects that are ongoing. And we've got a wide variety of folks in terms of their role at the institution, a lot of early career folks using the data, as well as even some fellows and teachers. And so it's really interesting to learn about the users who are coming on to use the resource. So just a quick note, if you were to want to get access to the data, it's quite easy. The first thing, there's a 5-step process. It does take a couple hours, but our goal is for this to be a few hours and not many weeks or months. I know some data sets take a long time to access. Our goal is that folks can join just in an afternoon. So the first thing to do, though, is to check for your institution's agreement. And the good news for University of Pittsburgh is that there is actually an institutional agreement in place. And so that means you've already passed step 1. And the next steps, the next 4 steps to register as a researcher, connect an ERA Commons account, complete the responsible conduct of training, and then sign the code of conduct. Those take about 2 to 3 hours of time, and then you can go in and start creating workspaces and running analyses. So we're really excited about that. And what's been really nice is thinking about the use of the data. And I know that under the leadership of Steve Reese and the Pitt Clinical and Translational Science Institute, that they've been thinking about a suite of resources that will help promote the use of that data within their institute. So I understand that the plans are still under development, but could include things like training and the relevant programming skills. I showed you a little bit about the analytics and the tools that are available in the researcher workbench. It takes certain skills, including the use of R and Python. So I know training in relevant programming skills and informatics tools, as well as guidance and expertise in study design. This is one of the things that we're really excited that we hope that users can sort of mentor each other, but it's wonderful if institutions can help support study design and data analysis. I know that they're thinking about potentially funds for pilot studies that would be selected through a competitive process and ultimately support on IRB proposals. So these are the sorts of things that we're really excited about, because we would love for institutions to get to a place where they're helping to support the use of the data here. We think that the All of Us research program data set could be a good starting point for many researchers, just in thinking about some of the questions that are hard to answer in the current data sets that exist. So that's really exciting for us. And I just want to spend a couple minutes on the last section here, which is our control tier that's coming out this winter. What we call our controlled tier, this is just another level of data. It really is about getting more and more granular. This is where the genomic data will live. We're also hoping to include more narrative data, clinical narrative data, data linkages, and it will allow us to share data that might have a higher risk of re-identifiability. It allows us to really get into connections between genomics and EHRs and all of the different types of data that are coming together. So we're really excited about this. We're expecting this to launch over the next 6 months, I would say. And what's expected in that launch is about 90,000 whole genome sequences. So of the participants that have joined our program, we've gotten through about 90,000 of their samples and generating whole genome sequence data. It's a huge amount of data that will be released and open to researchers. Ultimately, the goal is to generate 1 million whole genome sequences. So we're excited about that. But to launch, we'll have about 90,000 and then 120,000 genotyping arrays. We'll also be able to include in this tier of data COVID diagnoses and surveys, so more granular information about the COVID-19 pandemic and more detailed demographic data. And I think data linkages and other connections that get at the community level and some of those important aspects of our communities that might affect our health outcomes. So we're excited to see this launch, and then over time it will continue to expand as we all continue to share more data. The access requirements for the controlled tier are virtually the same as the registered tier, except there's a slightly different training and data use code of conduct to sign. But getting in and getting approved for controlled tier will be generally the same process as for the registered tier, all online at researchallofus.org. So with that, I think I will take any questions. And thank you again.
Thanks, Steph. That was fantastic. If you want to stop share screening, then
Yes.
we can move to questions. Maybe there's some questions already in the chat, but maybe I can take the first opportunity. One, to thank you for the talk. Normally there would be clapping, but you can't even do this on webinars. So I will clap for everyone. And I would say on behalf of everyone, it's amazing, you know, what you and the team and everyone has achieved. The, you know, setting the team up, the accrual, the diversity, and then I would be most excited about the genomics because that's what I do. So yeah, really fantastic. And maybe I can just take a first couple of questions. And so when I teach precision medicine, I always mention this every single time, and I tell people they should be enrolled, etc. And I'm always asked questions, and I taught it last week, and I was asked a couple of questions, so maybe I can just pose them to you.
Yeah.
The first one is around data breaching. So obviously, big risk when you have this huge data set of personal information in one place. And I presume you have state-of-the-art security, but even the best banks get broken into and stuff. What briefly could you say about that?
Yeah, well, no, thanks for that question, Adrian. It's definitely the thing I think that we, you know, worry about and think about quite a bit, right? We're endlessly devoted to protecting the data of our participants. So this is, of course, the thing that we spend a lot of time on. We have really incredible security teams who have spent a lot of time developing all of the various system protections that we need in place, including, you know, really robust monitoring and penetration testing and all sorts of things that need to be done in order to protect the data. We try to be as transparent as we can with our participants in the consent process and in communications with them, that our goal and our every attempt and, you know, an incredible amount of investment is going into keeping their data protected. And in the world in which we live, there's always a risk of breach or privacy incident or security risk, breach, or, you know, incidents involving private information. And that, you know, anytime that anything like that happens, we will be transparent and open about it, and we will do our best to mitigate any risk for them. So I think, I wish the answer could just be, well, we've got a foolproof system that can never be breached, and, you know, so we don't worry about this in one bit. But of course, it's a thing we always worry about. The other thing I might say is that we've held ourselves to a really high standard on the security front. We have what we call this process where authority to operate. It's based on a number of standards that the government uses to protect data like this, and we not compelled to go by it, but we have held ourselves to those standards, even though we really didn't have to, because we felt like those were really good and important safeguards to have in place. So we've got, I would say we've got an attitude of, you know, diligence, high investment in security and privacy, and then transparency if something does happen with our participants' data.
That's great. And one other quick question. Melinda Massart is here. She's in family medicine. She's fantastic, and she runs our precision medicine program, our clinical program. And I teach with her, and she presents this interesting case last year of a person who did ancestry testing and then took their data, ran through a third-party software, found these deleterious variants, which were not great. Turned out they were all wrong when she went on for clinical testing. How are you guarding against people doing, I know people, doing bad things with the data. I know you have obviously the regulatory entrance, but is there something to stop people when they get to their own data of them finding things by themselves?
Yeah, that's such a good question. I wish we were in a forum where we could just sit down and talk about this. You mean Melinda? Was it Melinda?
Yeah.
Yeah. So, that's such a good question. Let me say a couple of things about that. The data that our participants get back currently is an ancestry report or a trait report. It's not the raw data at this point in time. Although we are really interested in making that available for folks if they want it in the future. It's just not a capability we have at the moment. I think, we also, with promising to offer health-related results to participants, my hope is that they would use that service and allow us to do that, that test in one of our clinically validated labs. We've got a really elite group of genome centers that are involved in this that do the testing. And then the report out by a genetic counselor. I'm hopeful that our participants will instead opt to do it that way, as opposed to taking a raw data file when we offer it and going and running it through analyses themselves. Although, what's to stop them? We do have, I'll say one more thing. We do have a genetic return of results consent, a specific consent around the return of results, because we decided early on we didn't want to require people to get information back about themselves, but we wanted to walk them through a consent process that would allow them to choose to. And part of that is, information for them about all of the different types of things we can and can't know about our genome, or things we don't know yet about our genome, and how that information will change over time. And trying to just, again, lean into transparency about how the genome works and what we do and don't know, and how that information, what we do and don't know, will change over time. And so hoping to offer at least that level of understanding for folks. But that's a fascinating.
Yes, no, I mean, that's why it's used as a teachable moment, because it's kind of, that's why we have a precision medicine clinic, to deal with a new era of patients doing their own research. Yeah, so it was, I was just interested. I know you're not there yet, but you will be there.
Yeah.
And, it's a new world of data sharing and data analysis.
Yeah. Well, and I think I'll remember that case study now forever. So when we get to the point of returning, I'll be like, hey, actually, I know of a woman at University of Pittsburgh who had an experience with, and we might think about how we communicate that. And
Steve, I don't know if you want to say something.
Yeah, so Steph, thank you so much for being with us today. Really appreciate it. Actually, I had two questions. One is, we've been focusing on sequencing and genomics for the last several years, and sequencing 1 million people is pretty daunting, plus the arrays. But what are the other plans, or is there a strategy for other plans for different kinds of analyses with the samples?
Oh, that's such a good question. Here's how I think about it. If Josh Devaney, our CEO, were here, he'd have all the thoughts on this. I think that we will probably, now we're having an internal program conversation, but I love it. I think that we will probably run some assays on all 1 million folks on our own, right? That we will support and pay for, and it will go into the central repository. And I imagine there will be a number of researchers who will want to access a certain subset of samples and run some assays on their own, right? In some sort of ancillary study. And we'll have to think about getting that information back from those folks. I don't know what assays we'll choose, because we haven't yet made those decisions, and you're right, we have been so focused on genomics, but it would be really interesting even to run some environmental assays, and I could imagine a whole host of things. I bet you have a wish list as well. What would you think would be most interesting?
Well, I don't know. I've been so focused on genomics, but I would love for our investigators here to get together and think about, as a group, what we think we would be interested in, and is there a mechanism to provide input into program? My second question you already answered. Could we get access to actual samples at some point? And it sounds like we can.
Yeah, definitely. We have developed out the policy by which that would work. And now we have to decide, now we have to sort of build in the processes for how we would actually make that happen in terms of getting samples out and shipped to folks and back and all of the detail around that. But it feels like that's part of the promise here, is that it's not just us generating data, but that ultimately other researchers from across the country can do some of that using the resources that we've built.
That's great.
Yeah, so in the chat, there's a question from Mike Bisic. He said it was a great seminar. Mike asked whether this will be made available, and we have recorded it and we place it on our YouTube channel. And his question, which you addressed, actually, I think he asked it at 4:20, and I think you addressed it at like 4:30, was he said, I've been a reviewer of 2 All of Us manuscripts already. Are you open for business for all researchers now? And I think you addressed this. You said there's already been like 16 plus publications. I guess, is it freely open for anyone to do any research and publish, or there's still limitations? You know, some consortia say you can only publish X when certain stuff has been done, or is people free to do their research and publish?
No, yeah, it's fully open, assuming you can get through all of the steps of the onboarding. It's fully open. You can run whatever analyses are there. I mean, some of the, as you know, as I just mentioned, there's, you know, some of the data is available and not others, right? We don't have any genomic data out yet. But as for what data is available, yeah, that's fully open at this point.
Great, that's fantastic. So if there are no other questions, Steph, I'd like to thank you again. And Steve, thanks for the intro and for pointing us to Steph for the seminar. It was really great to have an update. Having seen it right from the start and Steve's group doing all of this, to suddenly now getting these answers, I think that's really great. There was a hands raised. So Richard Boyce raised his hands. Richard, if you could, I can allow you to talk. I have never tried that before. I could always allow. Richard, if you want to ask your question, you can.
Can you hear me okay?
Oh, yeah.
Okay, great. Thank you so much for your great talk and for all the great work you guys have done. I've been following this project since you all started, and it's evolving really, really well. My question is really basic. You did mention about training. I teach a foundations in biomedical informatics course, and I always encourage people, it's a project-based course, to check out all of us and get onboarded with that. One of the problems that people face is kind of knowing really what's in that data set and if they can answer their question that they want to ask. I know there's some characterization tools in there, but my question is, what do biomedical informatics researchers do to kind of partner with you and maybe look at other tools that could be built to characterize the data and answer certain questions, certain kind of data search, maybe algorithms and things like that we could try out? Is there any mechanism that you guys have thought of that would allow that to happen?
Yeah, thanks for the question. And I'll also let Steve jump in here too, because he and I were just talking the other day about some of the things that I know he's thinking about, just even at Pittsburgh. We have gone to some extent, we have developed some trainings around the tools that are available on the workbench, and the Data and Research Center that has built that environment and organizes all of the data and manages it all does a certain amount of support on the use of the tools and some trainings. I think, however, there's a ton of opportunity here for, as we build out our relationships with researchers across the country, in thinking about how others can sort of build trainings on top of the data, use the data to help mentor others, meet in the data, meet in a workspace and share expertise. So I would say we've barely scratched the surface of this at this point, as we've just released the data and we're starting to get to know how it works and how the tools work and all of that. And then I think Steve should talk about the training piece too, from his vantage point at an academic institution and collaborating with the CTSI. But on your point about analysis and software tools, we're very interested over time in building out our software capabilities. And I think there's a real opportunity to help to work with others to build tools that could be used within our cloud environment. But where we are now is just with the tools that have been built to start.
Yeah, if I can add, so thanks, Steph. So those are great questions. Richard, I would, for the high-level sophisticated tools, I would point you to Sham, who's a co-PI on our project here in Pittsburgh, who's very much plugged into the informatics community and all of us out of Columbia as well as in Vanderbilt. So he can probably help answer that question from a technical standpoint. From a trainee standpoint, we are putting together a suite of courses, some existing and some new, in your department, as well as the Institute for Clinical Research Education, to train our junior investigators in R and Python and more of the basic skills that are needed to access the data.
Okay, thank you.
Yeah, no, that was a great question and a great ending. I do think the hope is that this is going to drive more research, more education. I think it will. I think it'll drive new ideas and new ways to look at the data. And yeah, I think this is going to be a great resource. So Steph, thanks once again. That was really a fantastic seminar, and I think it really invigorates everyone to keep on doing the precision medicine research.
Thank you very much, Adrian, to you and to Steve and to everyone. Thank you.
Thanks, everyone. Bye-bye.